Dear students, welcome to Tungala online classes. So in the last video we were discussing what are nucleic acids, where they are present. So what do they do? I mean a function of these nucleic acids and how these nucleic acids are forming. So those simple structures that we discussed as well as chemical composition even we discussed. So those simple structures are nucleosides, nucleotides that we discussed of both DNA as well as RNA, and in this video so we'll discuss thoroughly how polynucleotide chains are formed. So before that one we must have a little bit more knowledge about the nucleotides. So these nucleotides are sometimes said as higher nucleotides. Why higher nucleotides these are called as? Let us see now. Higher nucleotides. What these higher nucleotides are? So higher nucleotides are simply said to be as these are the nucleotides which are containing more than one phosphate group. See here, so higher nucleotides. See here, already you know DAMP, you know. DGMP, you know, DCMP, you know, DTMP, you know. Phosphate group is monophosphate here in all these one. Only one phosphate group is present in all these one. But if it is more than one, then what is the condition? This becomes two phosphate group, then ADP, diphosphate. Diphosphate in the sense that two phosphate group. So higher to this one, DGDP. In the same way, higher to this one, DCDP. In the same way, so DTDP. Adenosine diphosphate, guanosine diphosphate, cytidine diphosphate, thymidine diphosphate. So this is a higher nucleotide, it is called as. This one is called as higher nucleotide. More than this one sometimes D, ATP, D, GTP, D, CTP, D, TTP. So deoxyribo, deoxyadenosine triphosphate, deoxyguanosine triphosphate, like this. So these are once again said to be as what higher nucleotides. So if the nucleotides have more than one phosphate group, they are said to be as higher nucleotides. They are said to be as. So this is also one more thing. So these are the energy sources. See here ATP is the energy source. See here ATP is the major energy packets or source in organisms, both animals as well as plants. And this ATP was discovered by Lohmann, right? So Carl Lohmann and Lippmann, right? F. Lippmann are the two scientists, the one who discovered this ATP, and they said that this is the major energy source in both plants as well as animals. This is the fact you must know. Apart from this one, let us go through the polynucleotide chains. Polynucleotide chains. What is this polynucleotide chain? Poly means many, nucleotide means nucleotides, right? Chain. Many nucleotides together form chains. So that is nothing but what? Polynucleotide chain simply. Poly means many, nucleotides, right? Chain. many nucleotides chain it is? Very simple one. So let us see one nucleotide. What makes one nucleotide? See here, one nucleotide is made up of, one nucleotide is made up of what? One pentose sugar plus one nitrogen base plus one phosphate group. Okay, all these three together form what? One nucleotide. This is the fact which is known to us, right? So let us see diagrammatically the same thing over here. So let us see diagrammatically. So what is this one? One pentose sugar. See here, this is one pentose sugar. So this is one pentose sugar. Of course, this is one pentose sugar. Okay, here we know carbon atom number 1, 2, 3, 4, and here fifth one that we come across. Here fifth one we come across here. Okay, this is one pentose sugar plus one nitrogen base. One nitrogen base must be there. Of course, I shall add here A. A stands for what? Adenine. One nitrogen base and one phosphate group on the fifth carbon atom. So there OH is deleted and there one phosphate group is added there. All these three together form what? All these three together form one nucleotide. One nucleotide is formed over here. Very simple aspect. So this is one nucleotide. Such kind of many number of nucleotides are joined over here. I shall draw the same thing. See here, this is another pentose sugar. Sorry. See here, this is another pentose sugar. This is also another pentose sugar. So these are the pentose sugars. Simply we can draw. See here on the first carbon atom, one more what? Nitrogen base is added. On the fifth carbon atom, there one phosphate group is added. Okay, and here also same, one nitrogen base, one phosphate group. This is one nucleotide, this is another nucleotide, this is another nucleotide. How to join these nucleotides? See here, one pentose sugar plus one bond is there between, between one pentose sugar as well as one nitrogen base, and this bond is called as what? That is glycosidic bond. So, and one pentose sugar, on the nucleoside, this became on a nucleoside, and phosphate group is there. There is a bond called as phosphoester bond. And one more bond is formed on the third carbon atom. So that bond joins two nucleotides over here, and this is called as phosphodiester bond. Means what? Here one nucleotide joins with another nucleotide with the help of a bond called as phosphodiester bond. What is that? Phosphodiester bond is present between two nucleotides over here. Very simple. Very simple. So there are three bonds that we come across. One is glycosidic bond, second one is phosphoester bond, and third one is phosphodiester bond. These are the three bonds you must remember over here, so as to know these nucleotides as well as polynucleotide chains. See here, this once again becomes what? One polynucleotide chain like this. It makes one polynucleotide chain. See, simply it is also a pentose sugar. See. And this is a T, and this is a P, and this joins to the third carbon atom over here. This is one polynucleotide chain. Right? So poly means many. Many nucleotides join together form one polynucleotide chain. Very simple one. Okay. And in DNA it is a double-stranded, I said. What do you mean by double-stranded? So this is one strand of DNA and the strand must be here. And the strand must be here. So we can draw that one also. See here. This is one nucleotide. Okay. This is one nucleotide. What I am drawing here. This is another nucleotide. What I am drawing here. Sorry. This is another nucleotide. This is another nucleotide. Okay. Once again I have drawn the nucleotides over here. This nucleotide joins to this one. This nucleotide joins to this one on the third carbon atom. On the third carbon atom. Right? This is first, second, this is third carbon atom over here. Like this. On the third carbon atom, the phosphate group joins to another nucleotide chain. So this is one strand of DNA, this is another strand of DNA. How many strands of DNA are here? 2 strands of DNA are over here. But both the strands of DNA are not joined each other. With what they have been joined? They have been joined with the help of the bonds present between these nitrogen bases. So A as well as the T are complementary to each other. Why I have written T only here is, so here one purine is always combining with one pyrimidine. Especially adenine combines with thymine, not at all other pyrimidine. It does not combine with C. So that's why these two are complementary to each other. They are said to be as complementary to each other. So this does not survive without joining to A does not survive without joining to T. So and both of these are joined with the help of the bond called as hydrogen bond. So how many hydrogen bonds these two are joined with? 2 hydrogen bonds. A combines with the T with the help of 2 hydrogen bonds, and G combines with the C. Means G as well as C are complementary to each other. So they join each other with the help of 3 hydrogen bonds. Once again here also CG, even though they are reversed, so they join with the help of 3 hydrogen bonds. They are complementary to each other. And here 2 hydrogen bonds. What it suggests? What it suggests is, so DNA is made up of two what? So polynucleotide chains. Right? Both are complementary to each other. Right? See here, one more thing here I have left over here. This is the fifth carbon atom it is. This is 5th carbon atom is left behind, and see here this is 3rd carbon atom left behind. Okay, the terminal parts you can observe over here. Here you can observe that is phosphate group is attached on the 3rd carbon atom, and here it is 5th carbon atom. So this is 5, this is 3 dash, this is 5 dash. This is 3 dash, this is 5 dash. Hence, so both the strands are anti-parallel to each other. So this is running from 3 dash to 5 dash here, and this is running from 3 dash to 5 dash, from base to upper part. So both are anti-parallel to each other, anti-parallel to each other, and both are complementary to each other. This is shown over here. Okay. Now I hope you got an idea how the skeletal structure of this DNA is. So right. So very simple aspect it is. So but once you understood, then it is always there in your mind, fixed, right? So if you don't once, then it is very difficult to understand, right? So that's why you must remember all these aspects over here. So what these DNA as well as RNAs are? These are nothing but heteropolymers of the nucleotides. There are different nucleotides that we come across. What is this nucleotide called as? T is there, pentose sugar is there, 1-phosphate group is there. What is the name of this? What is the name of this nucleotide? That is deoxythymidine monophosphate. This is deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxycytidine monophosphate, deoxythymidine monophosphate. So like this, the names are okay. So now I hope you have understood the structure of this DNA. So that is, so thoroughly I go through the ultrastructure of DNA, so which was made by Watson and Crick. So the Watson and Crick model of DNA will discuss now itself, right? So I hope you understood what are nucleosides, what are nucleotides, and what are polynucleotide chains, right? So these three things, if you know better, then it is very easy to study these nucleic acids over here. So let us start with the structure of DNA. Now let us start with the structure of this DNA. Structure of DNA. Let us study the structure of DNA. Simply, so this is the structure at molecular level I have drawn. Simply how the DNA is looking to be. So that I am going to draw over here itself. So this is how the DNA is seen. Okay. So it is double-stranded structure. So this is 3 dash. Why 3 dash is there? And this is 5 dash. This is 3 dash. This is 5 dash. Means anti-parallel to each other. And you will come across to this kind of horizontal ladder with joints. What these are? Let us see. See here. If A is here, double bond T is here. If G is here, triple bond C is here. If C is here, triple bond G is here. If T is here, double bond A is here. If A is here, double bond T is here. Okay. So this is how the structure is made up. And one more thing. See here. This is P, this is S, P, S, P, S like this. Okay, this is the backbone of DNA, what we call it as. Okay. Okay. So whenever we go through the structure of this DNA, there we find this kind of double-stranded structure of DNA. So how do you explain this one is the major aspect. See here, this is 5-mark question it is. So very important one, you must go through this diagram as well as So explanation very clearly. So let us start with the structure of DNA. See here, Watson and Crick are the two scientists, the one who made this model of DNA for the first time, and hence this model of DNA is called as Watson and Crick model of DNA. So Watson and Crick model of DNA is called as. This is first point to be noted over here. Second point. And this DNA structure is made up of two polynucleotide chains. So DNA is made up of two polynucleotide chain. Okay. DNA is made up of two polynucleotide chains, right? Two polynucleotide chains they are made up of, and both the polynucleotide chains are coiled together, forming a double helical structure. Both polynucleotide chains coiled together, forming a double helical structure. Hence the model of DNA is called as Watson and Crick double helical model of DNA. It is said to as Watson and Crick's double helical model of DNA is called as. Why? Because both the polynucleotide chains coiled each other, right? And so forming double helical structure. And fourth point. What is the fourth point here? Both the polynucleotide chains are held together with the help of the hydrogen bonds present between two nitrogen bases. Both polynucleotide chains are held together with hydrogen bonds present between two nitrogen bases. A always combines with D and C always combines with G. So here what it suggests is both the polynucleotide chains are complementary to each other. Already I have mentioned this one. Both are complementary to each other. Why? Because A always combines with the T. In the same way, G always combines with C. Right? So this is why we say that both are complementary to each other. Point to be noted over here. Right? Both the polynucleotide chains are held together with the help of the hydrogen bonds present between two nitrogen bases. And nitrogen bases are complementary to each other, hence both the polynucleotide chains are complementary to each other. And the backbone of this DNA is made up of phosphate as well as sugar. Backbone of polynucleotide chain is made up of phosphate as well as sugar and phosphates. Okay, next one, this DNA structure, both the strands are here, and both are complementary to each other, and it shows that bases are equivalent to each other. So basis of this strand is always equivalent to the basis of this strand. And this was first said by one of the scientists, right? So Chargaff is the name of the scientist. So Chargaff's base equivalence rule he made. Let us write Chargaff's base equivalence rule he made. What according to his rule? Number of purines are always equivalent to number of pyrimidines. He says number of purines are always equivalent to number of pyrimidines. Okay, this is the fact what he said. Means if how many number of A's are there, they are always equivalent to number of T. How many number of G's are there, always equivalent to number of C over here. Along with this one, he said A plus T divided by G plus C is always equivalent to 1. So this is also said by himself. Okay. Number of A are always equivalent to number of T. Number of G are always equivalent to number of C. Okay. AC are purines, TC are pyrimidines. Number of purines are always equivalent to number of pyrimidines. So based on this, some of the questions are rising in a NEET examination. So say for example, he says if A equal to 30%, if A base is 30%, what about all other? What about the percentage of all others, he asks. Then how do you answer? See here, A plus T plus G plus C is equal to 100%. It is always equivalent to 100%. Remember, this formula must be there in your mind. Okay, if A equal to 30%, A is given. A is always equivalent to what? T. If A value is given as 30%, then what about the T? It is also 30%. What happened now? A plus T is equivalent to 60%. They covered 60% over here. Remaining G and C. What about these two? That is 20 plus 20. Right? That is equal to what? Totally 100%. It is 100%. This is how some of the technical questions are usually asked in the examination. That points to be there in your mind. So this is about Chargaff's base equivalence rule, he says. Number of purines are always equivalent to number of pyrimidines, he says. Okay. So this base equivalence rule you have to mention in the structure of this DNA. This is the fact. And one more thing. The distance between two polynucleotide chains. See here, this is the distance between two polynucleotide chains. It is about 20 angstrom units. How much it is? 20 angstrom. So the distance between two polynucleotide chains is 20 angstrom units. Okay. One complete turn of DNA is called as. See here, this is one complete turn of DNA is called as. And this is called as guide. What it is known as? Guide. One guide is equal to 34 angstrom units. One guide is equal to 34 angstrom units. One guide consisting of 10 base pairs. One guide consisting of 10 base pairs. What are these base pairs, you may ask? So this is one base, this is another base. So they paired together. This is one base, this is another base. Paired together. So this is one base pair, this is another base pair. This is another base pair. This is another base pair. How many base pairs are here? 5. 1, 2, 3, 4, 5. Here also 1, 2, 3, 4, 5. So here to here, total distance between. So what? One complete turn of DNA is called as one guide. One guide consisting of one guide equal to 34 angstrom units. The length is about 34 angstrom units. What about one base pair? The distance between one base pair itself is 3.4 angstrom units. Distance between two base pairs is equal to 3.4 angstrom units. 3.4 into 10 is equal to 34 angstrom units. It makes one complete turn of DNA. It makes one complete turn of DNA. That point is also mentioned here. One complete turn of DNA is called as guide. One guide is equivalent to 34 angstrom units, and one guide is consisting of 10 base pairs, and the distance between two base pairs is 3.4 angstrom units. In case of normal right-handedly coiled DNA itself. Right? So what is this right-handedly coiled, what is left-handedly coiled DNA? So there are different types of DNAs that we come across. So some are artificially meant, some are naturally occurring one. So all those things later, so we'll discuss in the higher studies, that is in POC2 we'll discuss. But in a normal DNA, right-handedly coiled DNA, so one guide is equal to 34 angstrom units. And it consisting of 10 base pairs. Sometimes 11 base pairs are also present in some other DNAs. Let it be, never bother about that one. This is the fact you must remember over here. So and one more thing. There we come across two grooves over here. See here, so this groove you are observing. See, this is the groove. This is the groove. What you are observing is called as major groove. Major groove it is called as. Why? Because it is larger and it is measuring about 2.2 angstrom units. And here you come across a small groove. It is called as minor groove. Minor groove, which is measuring about 1.2 angstrom units. 1.2 angstrom units it is called as. Right? So major groove, minor groove, and then respective measurements you must understand over here. So this is how the structure of DNA is. All these 11 points or 10 points you have to mention compulsorily, then you will get 5 mark. This is one 5 mark question. Okay, so many 1 mark questions are also asked over here. Right? So so different types of DNAs I said. One is Z DNA that we have come across, B DNA that you come across, C DNA you come across, right? And one more is circular DNA, right? Circular DNA, mitochondrial DNA, chloroplastic DNA, all these are different types of DNAs that we come across. And even palindromic DNA is also one more type of DNA is there. So we need not to worry about all these different types of DNAs. So we shall go through different types of DNA along with RNA structure itself. Now we shall go through different types of DNAs now. So first DNA is B DNA. It is a right-handedly coiled DNA. It is right-handedly coiled DNA. So in which usually, so as we know, how many number of base pairs per turn that we come across is, so here 10 base pairs per turn that we come across over here. And helix rise, right? Here base pairs what we come across, base pairs what we come across, and helix rise per base pair. Helix rise per base pair is 3.4 installments in this case. It is natural DNA, right? And next one is aDNA. aDNA is also a right-handedly called DNA itself. It is also right-handedly called DNA, and this aDNA has base pairs is about 11 number of base pairs that we come across each term, right? In each term 11 number of base pairs. Helix rise per BP itself is about 2.6 installments over here. And one more, third one, zDNA. It is left-handedly called. DNA is left-handedly called DNA, whose base pairs is about 12. Okay, and helix rise per BP itself is 3.7 installments. Right? This is the difference between all these three types of DNAs what we come across. Along with this one, some more types of DNAs are there. Next one is circular DNA. That is cDNA, it is also called as, that is plasmids. Plasmids are also kind of DNA, extracellular self-replicating, right? Circular DNAs, these are commonly known as, I have already told you many times during the study of the bacterial cell, right? So these are extra chromosomal self-replicating circular DNA is present in bacterial cell. They are circular DNAs, what we call them as. One more thing. So next one is, after the circular DNA, so mitochondrial as well as chloroplastidial DNAs that we come across. These are also circular DNAs that are present in the cell organelles. And one more type of DNA is palindromic DNA. It is called as. What is this palindromic DNA, you may ask? See here, on DNA strand there you come across the bases. Okay, so I will draw over here. This is double stranded DNA. It has two strands. One is running from 3' to 5' and another one is also 3' to 5'. Both are anti-parallel to each other. On this you will come across the bases. See here, these are the bases. So if it is A, this is D. If it is G, this is C. If it is C, this is G. So like this, T, A, double bonds that you come across, triple bonds, triple bond, double bond, and here A double bond T, like this basis that you come across here. If so, the DNA in Hodge, so M-A-D-A-M, madam. So when you read this one in both directions, then the spelling is same. So if the bases are arranged this way, then that kind of DNA is called as palindromic DNA. Okay, palindromic DNA is called as. So R G A D A Z Gadag. So you may read in all the directions, in both the directions, then it is similar. If the bases are arranged like this, then such kind of DNA is called as palindromic DNA. Its use is more in case of, so that is plasmids. So there the one new DNA strand or the new gene is introduced into this plasmid, any one of the plasmids. So that is rDNA technology. You might have heard of this one. So in rDNA technology it is used vastly, right? So there palindromic DNA you will study thoroughly there yourself, right? So these are different types of DNAs that we come across over here. Along with this one, RNA is also more important one. What is the unique features of RNA? Let us see in this video. In the next video we will see the different types of RNAs there. Okay. Yes, next one is, next nucleic acid is RNA. This is a ribonucleic acid it is called as. And so let us see this one. So RNA is another type of what? The nucleic acid. It is the second type of nucleic acid, and this RNA is so differing from DNA. So it is differing from DNA because of the presence of the pentose sugar. That is, ribose is the pentose sugar present in this one. But in DNA, so deoxyribose is the pentose sugar present there. Apart from this one, nitrogen bases, phosphoric acid, both are same. Since phosphoric acid is there, they are acidic in nature, already have mentioned. Right? So this RNA that differs from DNA because of this pentose sugar, presence of pentose sugar. And this RNA occurrence. Where does it occur is the question arises. RNA, so sometimes act as the genetical material and sometimes it is acting as non-genetical material over here. So it acts as genetical material. It acts as genetical material in some organisms. Okay, so such kind of RNA is called as genetical RNA. Okay, this is the different concept. So RNA is present inside the nucleolus. So this is also present inside the cytoplasm. Right? That is inside the ribosomes. In cytoplasm it is present inside the ribosomes, right? So this way, this RNA is occurring there. This RNA is also the genetical material of some viruses called as retroviruses. Retroviruses are those viruses. The best example is HIV, human immunodeficiency virus, is the best example for retrovirus. Why? Because this virus consisting of a reverse transcriptase enzyme here. What it consisting of? A reverse transcriptase enzyme consisting of which help in the synthesis of viral DNA, viral RNA like this. So that's why, reverse transcriptase enzyme, since it is present inside these viruses which contain genetical material as the RNA, so they are called as retroviruses. Retroviruses, these are called as. Okay, so this is the occurrence of this RNA itself, right? So this is the occurrence of this RNA. And let us go through the types of RNAs. So there are major two types of RNAs that we come across. One is genetical RNA and the second one is non-genetical RNAs. So there are two types of RNAs that we come across. One is non-genetical as well as genetical RNA. If the RNA acts as genetical material, if it acts as genetical material of a particular organism, then it is called as genetical RNA. Non-genetical RNA is the RNA which is not acting as the genetical material, not acting as genetical material here. So, and these non-genetical RNAs are once again classified into different types. One is mRNA, another one is tRNA, and another one is rRNA here. These are the different types of RNAs. So they are non-genetical in nature. Why? Because these are not acting as the genetical materials over here. Hence these are called as non-genetical RNAs. We will study these types of RNAs in the next video thoroughly over here. So thank you very much.